Activation of the beta-adrenergic receptor by epinephrine or the glucagon receptor by glucagon results in the activation of adenylate cyclase, which produces the second messenger cyclic AMP in the cytoplasm. Cyclic AMP in the cytoplasm will activate protein kinase A, pKa, which in turn will phosphorylate specific enzymes on serine and threonine residues and changes the activities of these enzymes. In some cases, the effect is to activate the enzyme. In other cases, the effect is to inhibit those enzymes. In glycogen metabolism, pKa can phosphorylate glycogen synthase, and in doing so, it will inhibit the enzyme and glycogen synthesis or glycogenesis will be inhibited. On the other hand, pKa will phosphorylate the glycogen phosphorylase kinase, which in turn will be activated and can phosphorylate glycogen phosphorylase and activate the latter enzyme for the hydrolysis of glycogen. Importantly, glycogenolysis in the liver produces glucose because the liver has glucose 6-phosphatase, and thus it can release that glucose to the bloodstream. On the other hand, in the muscle, glycogenolysis can only produce glucose 6-phosphate, which can be used in glycolysis to generate energy for the muscle. But it cannot be metabolized further because the muscle lacks glucose 6-phosphatase. And therefore, it cannot supply glucose to the bloodstream. The rate-limiting enzyme responsible for the degradation of glycogen to produce glucose 6-phosphate is glycogen phosphorylase. In the liver, this enzyme is allosterically inhibited by glucose 6-phosphate, and ATP. After a meal, a rise in glucose 6-phosphate, and ATP will allosterically inhibit glycogen phosphorylase, and therefore glycogenolysis will stop. Simultaneously, a rise in glucose 6-phosphate in hepatocytes will activate glycogen synthase, and resulting in an increase of glycogen synthesis in the liver. Activation of the beta-adrenergic receptor by epinephrine or the glucagon receptor by glucagon causes a rise in cyclic AMP, which in turn activates pKa, protein kinase A. One such enzyme that's phosphorylated by protein kinase A is phosphorylase kinase B, which is then converted to phosphorylase kinase A, the active form. Phosphorylase kinase A in turn phosphorylate glycogen phosphorylase B and convert it to the active form glycogen phosphorylase A. Glycogen phosphorylase A will then be able to hydrolyze glycogen to produce glucose 6-phosphate in the liver. Alternatively, epinephrine through the alpha-1 adrenergic receptor will activate the release of IP3, which in turn will mobilize calcium from the ER, and the rise in intracellular calcium will then bind to calmodulin and allow this molecule to activate glycogen phosphorylase kinase B without phosphorylation. Then glycogen phosphorylase kinase A will phosphorylate glycogen phosphorylase B, converting it to the active form glycogen phosphorylase A, which is then able to hydrolyze glycogen to produce glucose 6-phosphate. The rate-limiting enzyme responsible for the degradation of glycogen to produce glucose 6-phosphate is glycogen phosphorylase. In muscle, this enzyme is allosterically inhibited by glucose 6-phosphate, and ATP, while it is allosterically activated by calcium and AMP. After a meal, a rise in glucose 6-phosphate, and ATP will allosterically inhibit glycogen phosphorylase, and therefore glycogenolysis will stop. During fasting and/or exercise, calcium inside the muscle will rise due to its release from the sarcoplasmic reticulum during exercise. In addition, AMP inside the muscle will be increased due to the consumption of ATP by the muscle. This rise in calcium and AMP will allosterically activate glycogen phosphorylase and stimulate the degradation of glycogen in muscle tissue. Activation of either the beta-adrenergic receptor or the glucagon receptor causes a rise in intracellular cyclic AMP, which in turn activates pKa, which phosphorylates several target proteins. One of the enzymes phosphorylated by pKa is phosphorylase kinase B, which converts this enzyme to phosphorylase kinase A, the active form. In turn, phosphorylase kinase A is able to phosphorylate glycogen phosphorylase B and convert it to the active form glycogen phosphorylase A, which in turn is able to hydrolyze glycogen to produce glucose 6-phosphate. Another pathway during exercise that results in activation of glycogen phosphorylase is the rise in intracellular calcium released from the sarcoplasmic reticulum during exercise. This intracellular calcium can bind to calmodulin and activate this molecule, which allows it to bind to glycogen phosphorylase kinase B, converting it to glycogen phosphorylase kinase A without phosphorylation. Bound to calmodulin, glycogen phosphorylase kinase A is able now to phosphorylate glycogen phosphorylase B and convert it to glycogen phosphorylase A, the active form of this enzyme, which then proceeds to hydrolyze glycogen to glucose 6-phosphate. If you like this video, please subscribe. Thank you.